you mentioned when you were, looking at all these ectotrophic, runner hyphae and all these different pathogens causing, take all root rot, that they weren't exactly looking the same. The hypophodia is a little different. That curiosity got to you when it came to discovering another new pathogen that your lab helped work on. And that is the pathogen that causes Inkspot, correct?
Correct.
What was that like from the first time you got a sample and you're looking at it? And I'm just trying to imagine, you're looking at a sample and you think, okay, I think this is a curvularia, which could take sometimes less than 10 seconds to confirm under a scope. Mm-hmm. And that didn't happen with that one. So could you kind of walk through the birth of Inkspot, if you will?
Okay. Well, it was a Friday night in the lab, and it was actually a 328 sample, and it had foliar symptoms that just didn't look like a dollar spot symptom, and it didn't look like a bipolaris leaf spot symptom. And I was like, what in the world is this? So I immediately removed some of the infected tissue, and we surface disinfest, and I just put it on water auger. And I thought, I'm going to let it grow out over the weekend and see what's there. But in the meantime, when I was looking at the plant, I saw a lot of very black hyphae down kind of in the stem region of the canopy. Mm-hmm. And I didn't see any sporulation or, sexual reproduction going on like you would if it was Curvularia lunata, mm-hmm. which, sporulates profusely. And I didn't see any of that. And I thought, well, this is pretty weird. So the next Monday I came back and there was this robust hyphae coming out, and it was clear and there was no sporulation, but within a day or two, it turned black. And I was like, what in the world is this? I had no clue. And I had to report to the superintendent, there's a foliar disease, but I don't know what this is. I've never seen this before. And so that was in 2007. And 2016, we were finally able to publish after years of characterizing the fungus and figuring out, where this fungus belonged in a genus. And it kept going into Curvularia. And I thought, well, it's just not sporulating. And I'd put it on multiple nutrient augers and put it in light, UV light, no light, 6 months in the dark. And it just never sporulated. Nothing. So the taxonomists, when we were publishing, they were very apprehensive because they said, well, it's Curvularia and it's not, sporulating. And there was a book, The Atlas of Turfgrass Diseases,
yes. Yep.
And it came out of Japan. And there is a Curvularia blight that they have a chapter of, and the pictures are exactly like the symptoms that we were seeing here, and I first saw it in Texas. And over the years, then I was seeing it on our research greens at Mississippi State, and I was very excited about that. But in this book, the causal agents were Curvularia lunata and several other sporulating curvularias, and then a little mention down below, and a sterile fungus was associated with it.
Yeah.
And I was like, I've got the sterile fungus. And I showed that it's, much more pathogenic than Curvularia lunata, and it, produced the same, symptoms. And so, it took a long time and, trying to break through and, show that, okay, this is a Curvularia, but it's sterile. I don't know why it's sterile, but, in some of the old writings of like Dreschler, when he was characterizing the Helminthosporium fungi, he talked about some are just sterile.
Yep.
And, you just have to accept it. And this unique fungus, which is highly pathogenic, and I think the most pathogenic curvularia in the group that are associated with diseases, our turf diseases, this is highly pathogenic, and it causes some pretty major damage in the foliar region of our greens. It's on fairways, it's on tee boxes. I haven't seen it in athletic fields or home lawns. Just, shorter mowed golf turf.
Yep.
And it likes a lot of water.